right, so good morning. Today we have the pleasure to have with us Dr. Simon Archibald, who is the Chief Scientific Officer of Integra and spent his career studying both spinal cord and peripheral nerve regeneration. I think this morning is going to give us a story of the development of intubulation or conduits for nerve repair. And so it's an honor to have you with us, and I'm going to go ahead and turn it over to you.
Justin, thank you very much for the introduction. So I'm going to give an arc of from sort of early work on intubulation, repair, and peripheral nerves to the work we're currently doing, which is, as the title suggests, is really sort of creating a bioartificial nerve graft. I've got a thing right in the middle, so I can do it from here. So it's Rama Cajal. And I think one of the themes of the talk is things we possibly should have known because people discovered them beforehand and really didn't understand what the significance was. Or that in retrospect, we see how significant the observations were, which is good. We continue to evolve. So this is a very classic picture from Cajal, and a couple of pictures really talk all about the things which we really care about in peripheral nerve regeneration is the chaotic regeneration when you put two nerves together, the amount of axonal escape from a nerve repair, which has a lot of other problems, in addition to painful neuroma, hypersensitivity of the skin. The other picture also shows us, the propensity of axons to actually grow in the right direction and find their way. And if you look very carefully at the drawing, you see a stream of cells connecting the two severed stumps. So here is indication of the Schwann cell involvement of bridging gaps between peripheral nerves. And this is all from just the beginning of the 20th century. So if you know what you're looking at, you can really see a huge amount of detail.
Now, on these images, obviously, we're not seeing the Schwann cells. We're seeing what the axons are.
Yes, but if you look very carefully in the background, he's drawn with a stream of cells.
It's dot. Okay.
Yeah. Yes, it's a silver stain for axons, which is very nice. You bring that forward a bit more, Tom Bruchart's work showing a very similar picture of axonal regeneration across a nerve gap. So you're adding some more late 20th century technology to analyzing what's going on. So for a long time, peripheral nerve repair, was not understood what was happening. Early part of the 20th century, all sorts of ways of sewing nerves together came along. And then people started to work on sewing nerve trunks together, getting the right orientation. But it's very interesting that all that work done by Cajal really didn't have any effect on how people thought about doing peripheral nerve repair. So we came up with a very mechanical solution to this very biologic problem, and you approximating the nerve stumps in different ways and suturing them together. And then the big advanced Malaysia I started using the microscope in the late 50s, early 60s. And that really did improve because people would actually see what they're doing very carefully. And now we use microscopes or loops. But a lot is going on. It is a very mechanical solution to this biologic problem. This is a rat nerve, tibial nerve, which has been repaired with a microsuture. And you can see how if you use a single suture, it's distorted. And if you look under the electron microscope, the scanning electron microscope, you see how much of the endoneural contents are bulging out, how much they're exposed to the environment, the opportunity for loss. So it's not, it's very good, but not as good as you could possibly get or envisage. Some people, Domenicelli, created a very slow cooling, which didn't freeze the nerve, but took the fat down so that you could actually cut it. It was mechanically stiff, so you could cut it without the bulge forming. And then you could approximate the two ends and the fibrin clot would sort of glue them together. And as you can see from the graph, it was actually a very significant improvement in the neurography by doing it in this very careful way. But it takes about 3 or 4 hours to cool them down appropriately without them dying and to warm them up. So it's not something that you're going to come into So a lot of people have looked at intubulation. There's French workers, late 1800s, early 1900s, used dog arteries to repair dog nerves and had really good results. And they used dog arteries to repair human nerves and had really terrible results and didn't understand why. We now know. But the concept, is quite good. Trachea of hens being used in the 1700s and other materials, pen quills, etc. All of which didn't really work very well, but the concept was out there.
So just for the audience in general, the dog nerves vessels not working because of the rejection of the information.
Yes. I made a gross assumption there. I'm sorry. And then some really early work by Paul Weiss in the 40s looked at the nerve gap over a period of time and started to understand what was going on. And he saw fibrin clot forming between the proximal distal stumps, and reorganization of the fibrin clot becomes oriented as it contracts. As the fibrin clot matures, it contracts. If it's between two objects, that tends to straighten it out. And it makes a very good substrate for nerve regeneration. And this is one of the things which really got forgotten for a long period of time. It's the fibrin clot. When you put a tube into a system which doesn't have anything in the lumen, it fills up with plasma exudate, fibrin clot forms, and it allows for a bridge. And what it's bridging is not axons, it's Schwann cells. And so this model, some of our work in the 90s, we actually ligated both ends and then looked at regeneration. You get this beautiful Schwann cell cable, bands of recreated. So you have reestablishment of the entire cycle architecture of the peripheral nerve without axons. So that changed how we looked at the system fundamentally. So we started looking not at axonal regeneration, but Schwann cell regeneration and promoting Schwann cell regeneration and migration, which is part of the end of the talk.
So can I ask a quick question on
You
that?
may.
Because the way you presented it sounds like the axons are going to go nowhere the Schwann cells do not exist.
That's correct.
They will not go if there's not a Schwann. Now, when we did neurotrophic factor research previously, we would see axons that would crawl across epineurium, or they would crawl in places outside of it.
They do, yes. But they're usually followed by Schwann. They're either, it's like who's on first.
Got it.
There's a Schwann cell, there's generally a Schwann cell there.
Okay.
If you're getting myelin, then there's definitely a Schwann issue, but the Schwann cells follow. But in the peripheral nervous system, so you can grow axons in petri dishes without Schwann cells,
right,
and they'll follow scratches. They do regenerate,
okay,
but the Schwann cells are actually the ones which are providing the pathway to the periphery, okay, and they're providing all the trophic support for the axons.
But in the setting of intubulation, if there's not a Schwann cell pathway, the axons are not going to jump from one side to the other. Okay.
I mean, you may get 2 or 3, but it requires a Schwann cell bridge. And the Schwann cells come out from both stumps. They form bands of Bugner, and the axons follow them down to the periphery. And then, so that work and some of the previous work, what the essential ingredients are, some sort of tube environment for these to occur in, the Schwann cells, and a matrix scaffold. Now, fibrin clot forms a matrix, but it's fickle. Autolysis of fibrin clot occurs about 8 to 12 days in humans, and it occurs faster as you go down the order of metabolic. The faster the metabolic rate of the animal, the faster the fibrin clot gets resolved. And that is entirely responsible for the different critical gap distances in different animals. So critical gap distance gets shorter and shorter as you go down through man, rats, mice. And it's the autolysis of the fibrin clot, because the Schwann cells are growing out of the fibrin clot, and they grow so far, fibrin clot liquefies, there's no substrate to grow on. So they then have to crawl, if they can, crawl along the wall of the matrix, the tube, and reach the other stuff, but that's much degraded. So that really fundamentally counts for the difference in critical gap distance. It's not the fact that mouse neurons are less able to regenerate than rat. But it was Paul Weiss really taught us that in the 40s, but it seemed to have got forgotten in the 80s and the 90s. So intubation was explored extensively in the 80s, particularly by Johan Lundberg in his work with silicone tubes. And it works very well over short gaps with the Fibrin Clot matrix within it. But the issue was that you had to have about 25% of the patients had to be reoperated to remove the silicone tube. They didn't actually use solid silicone tubes. So it was a silicone tube with a slot in it and it was sewn shut. So there was an ability for it to extrude if the sutures broke, or to be retrieved quite handily without damaging the nerve inside. They had very good regeneration. This is their technique. And they had their fundamental result was a dramatic drop in cold intolerance at the operative site, which really indicates that there's a very reduced amount of axonal escape. And subsequent studies show there's a reduced amount of neuroma formation and tubulation because you're having guided tissue regeneration, not getting the scar formation, and also the axons are directed into the distal pathway.
As a biomaterial, I understand that silicone is something that axons sort of don't like. They like to avoid it.
They hate it.
Yeah. Okay.
So silicone is not a really friendly material. It is, and this study shows exactly that. So you use a silicone rod. Lundberg used a silicone rod implanted subcutaneously to make a mesothelial tube or pseudosynovial tube. So it's a sterile abscess. The body is trying to ward off the silicone tube. It just doesn't like it. However, it makes a very nice model for regeneration. It makes a living conduit. And the level of regeneration is much improved over a synthetic impermeable material, as you can see in their results from the cross sections. So I started my career in 81, and so those were some of the papers I was reading at the time. We recapitulated some of that work in Kiel and at University in the UK. And this is our pseudosynovial tube, which you put a piece of stainless steel wire around, like Lundberg did, to keep the lumen open, otherwise it collapses. But you get this very nice sterile abscess, and it has a lot of properties like the blood-brain barrier. So it's very, will provide a nerve, blood-nerve barrier, etc.
So the tube is made of?
Tube, the inside, this rod is silicone. So you're creating an external tube.
So you're letting the body sort of develop a little scar sheath around it. You've got it.
And then you pull the tube out and then you take it off and use it to repair the nerves. And it works very, very well. However, you have to wait a month or so for the body to create the tube, and which took us to Harvard. And with Dick Sidman and Roger Madison, we started working on a number of different types of intubulation systems. But the collagen tube was the one which most matched the pseudosynovial tube, and it reanimated after implantation. So concurrently, Young at MIT was working on the skin graft to regenerate skin. So there was a lot of communication between MIT and us at Children's Hospital.
So when that pseudosynovium is formed on the silicone tube, does the negative aspects of the silicone tube go away? It's now encased in something that's
yes, it's a sterile abscess. Yeah, it reaches a quiescent point. Okay. So it's like as if you had a splinter in your finger. It'll have it'll be, if it doesn't get infected, it'll be encapsulated and then just sit there until it works its way out or whatever. It just is quiescent.
Except that it would remain impenetrable to vascularity.
Yes.
So if you get the tube out, this stuff probably will allow it growth.
Yes. Okay. So we work very on the porosity. So a lot of people make solid collagen tubes, not particularly porous. And we then found that, if you make tubes which had a porosity of about 68 angstroms to allow bovine serum albumin to freely cross, also a large number of growth factors are in the same size scale. That produced the best regeneration. And you can see from this very early rat work for the EMG that we're getting one of the tube candidates, the 68 angstrom tube, was performing as well as the Graft in that model at a 6 millimeter nerve gap, 4 millimeter nerve gap. So a very short gap, but it's a bioassay, not proof of regeneration. So you end up with this material, which is a spun collagen tube, which has a molecular cutoff membrane on the inner lumen and a supporting structure on the outside. And we were then had a series of grants to look at its function. One of the critical issues of tubes is that they must maintain their lumen over a long period of time. It's one of the huge drawbacks of using everted vein grafts is there's no structure and they collapse, and so that your cast of the fibrin clot within the material is a cast of the shape of the tube. So if it's a collapsed vein, that's what you're going to get. So if you have a tube which actually binds to collagen, so fibrin clot actively binds and cross-links to collagen. That's part of its evolutionary function. It doesn't do that to plastic. So in the plastic tube or polymer tube, it shrinks, as Paul Weiss showed us, and you get this classic hourglass shape. Is what everybody thinks about intubulation. You have an hourglass-shaped nerve connecting the proximal and distal stumps. Well, that's with polymer tubes. With collagen tubes, the fibrin clot is kept open and you have full caliber regeneration without hourglass shape. Unless, of course, you're beyond the critical gap distance and you'll see a thin ribbon where the fibrin clot liquefied. And you can see from this, there was a lot of technology and art in creating a collagen tube, which was basically a perfect elastomer. You can sit it in an instron and you can sit and run the instron for days. It will rebound to the same shape. If I'm being dramatic, I can put it on the floor and grind it into the floor with my heel and drop it back into the water or pop back into the tube. It's very tough and very resistant to compression, yet very easy to handle and suture. So we then had a series of grants. Integra for the first one didn't exist. It was about Colatech, and we collaborated with them on a primate model, looking at median nerve repair in the just distal to the crease in the wrist. And we followed these animals for 4 years or more, first group, and prepared tubes to graft to direct suture. And very good results all round.
What was the size of the gap and length of the tube?
It was 5 millimeters. So we now assume that that was good, but we didn't know at the time. And so our short gap neuropathy, we were done. This proved to be a very good device. And but as always, you continue. The data showed that we had supernumerate axonal regeneration in the distal stump compared to the normal population. It's what you expect. It's slightly more in a tube. You've got 2 points of branching. You have where they exit the proximal stump and where they enter the distal stump. So it's slightly more. All other factors show that you have a good and robust population of axons in the distal stump. If we take it on and look at the larger gaps, so this is the work that comparison from graft and tube. And this was really quite surprising to us to have this data in a very controlled environment, so sharp section injury and repaired with 3-stranded saw nerve graft. And you see that there's a very little difference except the delay in regeneration between a 5 millimeter graft and a 20 millimeter graft, but then a very dramatic drop in functional outcome according to the EMG at 5 centimeters. So really something which is in the general realm of what we use in surgery to repair nerves. There's a very definite decline in function with gap distance. And one of our conclusions of all the primate work and all the work we did is longer gaps do worse. which is, but it was really.
You could have saved your millions.
But it was really fascinating because then it really tempered how we were looking at what we were achieving. So there is a significant reduction between 5 millimeter and 20 millimeter in a tube, but it's better than a 5 centimeter graft. So then you have to ask as a surgeon, in a particular case, do I take, I've got a 20 millimeter graft, that's going to be better than a 5 centimeter graft, which I would normally do. Is that okay for that? And there may be sort of ancillary nerves, which that would be just fine, which you don't have enough grafting material to use. And so it was a sort of very philosophical point about how you choose the type of repair that you do.
Was this an autologous graft or?
Yes. Yeah, it's just the animal's own sural nerve being used as a repair. And previously, but this is tubes. And we thought that at 5 centimeters, we would have no recovery at all.
Right.
And we see a detectable EMG response in all the animals. So that, if you look at the data, look at the harvested material, you see about 1.5 centimeters of good growth from proximal and distal, and then a little ribbon of tissue connecting the two.
Hourglass looking.
Well, it was like full caliber ribbon.
Yes.
Because the fibrin clot had collapsed, and then you get a ribbon. So that's, but we then, I'd taken a sabbatical to be at Integra to take the technology into the clinic. And I'm on a 25-year sabbatical at the moment, hopefully to go back. However, we took this and did a randomized prospective clinical study with a lot of people who were very interested in peripheral nerve in Barcelona, headed up by Javier Navarro, and in Copenhagen, headed up by Christian Krara at Rigshospitalet.
Can I ask one question at last before we get into this? The fibrin clot that's collapsing in the middle, I assume it's because it's not generating enough to fully fill the tube. If you fully filled that 5 centimeter tube, would you then get more axon crossing? Is it a volume of fibrin clot?
It's the fibrin clot just liquefies. There's no substrate for the Schwann cells to grow on. So they have to grow on the wall of the tube. That's the only substrate available. And that's a 2-dimensional structure instead of a 3-dimensional structure. Right. So the amount of space is highly limited. And so that's why you get this thin ribbon connecting the two. But if the wall of the tube is not permissive to cell migration, then you don't get any regeneration at all.
You suck it out, put something else back in there.
Well, there's been work where you use metalloproteases to slow the fibrin clot formation, and that does help. It extends the life of the clot. And so you can do that. You can harvest some of what you can make your own plasma clot and put that into the tube. So you can mix it with metalloprotease and put it in the tube. It's been done experimentally. Tarloff was at Harvard and wrote a little monograph on the plasma clot, how to make your own plasma clots in the 50s.
You can't keep replenishing it.
No, because you're going to interrupt all the regeneration. So we have we thought about that, and we'll talk about it in a moment. So we did 32 patients and followed them for 2 years, and it's probably not so interesting. Sensory recovery, so the model is sharp section injury of the median arm and the lower third of the forearm. You prep it for surgery, and then you open the envelope, and it tells you whether you do a tube or a direct repair. So every single one could have been a direct sutures without tension. And so it was either direct suture or a 6 millimeter nerve gap.
This is the primate model again?
No, this is humans.
It's humans. Okay.
It's a multi-center randomized prospective study. And so at 12 months sensory and 24 months, you've got the conventional equals, suture equals conduit. Ulnar recovery was greater than median, as you might expect. At 12 months, motor recovery, conventional was slightly better, but not statistically better than conjugate, and they equaled each other at 24 months. And median was better than ulnar, which everybody would expect, but it's nice to have that under very controlled conditions. So that the EMGers were blinded to the procedure, and this is the ulnar recovery, and you can see that the two groups basically mirror each other, and the median nerve the same. The two groups mirror each other. So it's not a huge gap, but it's quite a significant gap, and there is actually no difference in the functional recovery, as you'll see also in the sensory side. But we recapitulated Joran's work and showing that there was a very significant reduction in pain at the operative site at early time points. And we showed that there was no significant difference between the functional tests of functional outcome tests. I think, at that point, we considered, we were done from the point of view of creating a neuroorthy assistance device, something which is quick, effective, preventive axonal escape. But really, the field is like, well, you can do 6 millimeters. I'm going to use it for 10 or 15, 6 millimeters. I'm going to do 10 or 15 millimeters. Everybody wants you to go on a bit further. And I think the field is generally forgotten that, we achieved the initial goal of making an improved neuroorthy system nerve connector. And then, this is a case by Michael Epstein showing, the utility of an intubulation in a difficult case. So this is an old 4-month injury, nice onion bulb neuroma on the proximal stump, and a very degraded distal target. And with just 2 sutures, you're able to match incompatible diameters, and the Schwann cells will sort it all right for you. Distal stump increase in size once it's re-innervated. So we are now looking at longer gaps and what do you do about longer gaps. And
once again, before you go on, I always have a question. When you do with the tube, is occlusive, occluding the ends of the tube helpful in a way? Does the sizing, does having ideal sizing matter?
No.
Okay.
So if you
there must be some parameters that
So when you section the nerve, it will start. Normal nerve injury, the nerve starts to swell. It can become inflamed. So you want to oversize the tube by a certain amount so that the nerve does not get trapped by its own swelling inside the loop. Because as soon as if it's swelling and becomes trapped, then you'll shut down vascularity. Okay. So that's fighting against you. And if you look at these implants, it doesn't matter whether it's a polyethylene tube or a silicone tube. You go back a week later, there's a thin film covering everything and sealing it off. You don't have to do anything to seal it off. Right? I have recommended over the years that you go 2 nerve diameters into the tube. So you go back, you go through all of the tube, you pick about nerve diameter back from the wall of the tube, you pick up the epineurin, perineurin here, go back through the tube and draw the vein into the tube. So now you're 2 nerve diameters in. If you just suture it onto the end of the tube, you have all the problems you have with normal nerve repair. You have capsizing of the suture line, and you have axonal escape. So you do need to be in the tube. Yeah.
And how much bigger should you go? Let's say the nerve diameter is like 3 millimeters. Would you recommend like a 4 millimeter?
Just one size up, another millimeter.
1 millimeter.
So, normal nerves, but the external matrix of Schwann cells is highly oriented and fills up most of the endoneurium. There's a few blood vessels in there as well. But we know that if you take the cells out of these, there's a huge number of ways of doing it. There's some commercial materials that you can then repopulate those pathways with Schwann cells, and axons will grow through. early work we did in collaboration with the folks at MIT was looking at using one of the matrices, candidate matrices for the artificial skin inside the tube, inside our tube, and then longitudinally frozen to make longitudinal pathways through the matrix. And that actually worked very well. It massively extended the critical gap distance in a rat up to about nearly 1.5 centimeters to 2 centimeters. The critical issue was making it. And so there's a lot of thermodynamics involved in getting the ice crystal to grow all the way through without deviating through conduit, which we finally figured out and worked the IP up on and allowed us to move forward. So as one of us you've asked earlier, one of the critical issues is, can you make it? And if you can't make it, then there's no way you could transfer it to the production or to clinics. So that begins to start to look like peripheral nerve. So you have the longitudinal matrix. There's a lot of collagen in peripheral nerves, so that's good. And this matrix has chondroitin sulfate, proteoblack, chondroitin 6 sulfate, proteoblack. Now we know from all our spinal cord work that proteoglycans tend to collapse growth cones. But this is not an axon regeneration system. This is a Schwann cell regeneration system. And it's in the peripheral nerve, it's chondroitin 4 sulfate, which collapses growth cones, chondroitin 6 sulfate. And you'll find chondroitin 4 sulfate limited to the wall of the perineurium, and it's a negative guidance cue. It stops axonal escape from the peripheral nerve. It's one of the things that prevents axons from growing straight out through all of the perineurium. So we have chondroitin 6 sulfate as our material. And so we worked with Alex Shin of the Mayo Clinic and his team. And this is a 14 millimeter nerve gap in a rat with a matrix filled tube. And you're looking at 12 weeks. So everything between the two sutures is completely regenerated nerve. So you have a completely regenerated nerve in that time. So it's the same caliber as for parent nerve.
The conduit is at both ends?
No, it's that's entire distance. The conduit went from here to here. This is new nerve from here to here.
Using you unwrap this. This is
No, the conduit was resorbed.
absorbed. Okay.
No sleight of hand. So one of the matrix filled tubes is implanted between the nerve gap. And when you're looking at the results 12 weeks later,
That's quite amazing.
it still makes me. So this is a nice, so these are the cross sections and
Of that piece of regenerated nerve.
yes.
Okay.
So which is in number 4, what you see at various types of preparation. So 1 is the nerve graft, 2 is just a collagen tube without any matrix in it, but you'll see that it's completely filled with regenerate. There's no space. 3 is a collagen matrix only, no chondroitin 6 sulfate. And 4 is the collagen chondroitin 6 sulfate tube. So you can see that, for a greater, from the point of view of the tubes, you're getting now, you look at the cross-section area and the density of the tissue, of the neuronal tissue, is very equivalent.
Again, the reddish color is going to represent axons or
And
Schwann
Schwann
cells. So number 2, you just get a little salt and pepper. Number 3, we have it centralized.
Yeah. And so if you look at higher power, you have dense, obviously, axons and Schwann cells, lots of microvesicles. The interesting thing between 2 and 3 is that you have the same clusters, but no microvesiculation. There's no perineural membrane around them. And 4 is the matrix-filled tube. And we had 20 animals, so we had 20 images from each group, so 18 when I went around with my stack of cards asking people to sort them out, and you got 2 groups of 20 and a group of 40. And that's
So the vascular is must grow longitudinally.
Yes, and it's very, very important.
So
you can
there's
see-
no radial ingrowth in any of these situations. It's all longitudinal. Coming with the
If
Schwann.
you look at the, now you see there's a lot of vascularity now on
Yeah.
the surface. So the graft is being supported by lateral vascularization. And in fact, if the tube doesn't resorb or you prevent collateral revascularization, you don't get regeneration. There's a study in cats with long gap tubes with electrodes in them around them with Christian Cryer. And if you don't punch holes all the way down and you crush, so you have a solid tube and you crush the nerve proximally, no regeneration occurs. You have to have collateral, which talks a lot about the quality of the nerve bed. And so, that's the number of axons. There's a lot of other data from this, but continue. So we have a tube, we have a matrix. How can we improve that? So the French Constant Group and James Fawcett at Cambridge in the mid-90s did a lot of work on the IgD epitopes of the laminin and fibronectin, and showed that it increased, those molecules dramatically increased the amount of Schwann cell regeneration from in culture. And so we recapitulated that with Fergal O'Brien's team in Dublin in 2D cultures of DRGs, adding various components of now, sort of 30 years later, you've got easy access to human recombinant molecules, which would make a clinical translation. And you take any one of these molecules and look in the culture, there's no change. You do not think that there was any significance. We've got laminin 1, laminin 2, and fibronectin. And as you can see, as you look at them individually on a collagen substrate, there's slight improvement. There's a lot of improvement over a straightforward lysine dish, but it's not interesting until you start putting more. And then you have this exponential increase with each additional molecule. So that you have in this one, you've got 1200 versus 2200, 800 versus 2200. So nearly a threefold increase in outgrowth. And then that is continued further if you go up to about 3,500 compared to 800 by putting 3 molecules on. And so there's a huge synergy in how these act. And you can see why a lot of people looked at these molecules in unitary, that they didn't see further, the reason why. And so that paper from French Constant really sort of spurred us to look at what the potential was. But it has, not only does it increase outgrowth and the rate of outgrowth that we just showed there, it increases the rate of proliferation, got a huge number more cells. But then the ratios in which these molecules occur in the substrate have just changed the response in equal amounts, sort of for NGF and GDNF. They don't really do very much. But if you make it laminin heavy, then you get this massive increase in growth factor expression and other growth factors. So just by changing the ratio of the substrate, you can induce the Schwann cells to change their behavior. And the work has taken us back to basic science to look at what's happening to the Schwann cells in the distal stump and how they're modifying their substrate and communicating to each other through the substrate to upregulate these components. And all of these molecules, there's these pro-generative molecules. So the VEGF and other angiogenic cytokines and substrates are incredibly upregulated. And so we used this model for looking at determining which the best substrate was and you can see that laminin 1 heavy, so it's 4 times the amount of laminin 1, so 1 to 4 to 1 was the candidate which we moved forward with and did the animal studies. And you can see that we have a very significant increase in angiogenesis in the tube by modifying the substrate. And the number of axons are significantly increased. But interestingly, it seems to slightly slow the progression. So we don't understand the fact that we get more axons. They're not quite as mature as the other axons in this beta model. So we need to do some more work about that. But having a sort of 40 to 50% increase in the number of axons in the distal target is a good start. Right? So the last bit really is cells. Schwann cells are so important. Why not put them in there? So the collaboration we've had with Alan Levy at Miami, going all the way back, he and I have known each other since he was doing his Ph.D. With Mary and Richard Bunge. And the goal has been to provide a human, autologous source or a compatible source of Schwann cells. So Richard Bunge's main idea, and a lot of work for the spinal cord injury, etc. And Alan's team has now achieved that. They have a method of taking autologous cells from debris around a nerve injury or from a sural nerve, etc., and massively expanding them and then maintaining their Schwann cellness and then putting them back in. And he has published clinically. And this is the ability to infuse the matrix tube with Schwann cells. So you put a matrix tube in the syringe and you just draw out Schwann cells into it. And they populate. We thought they would all end up at one end, but they sail through and attach throughout the length of the tube. So this is just after infusion. And then if you take that and you implant it, you have beautiful regeneration and a very significant improvement over the collagen matrix over a long gap distance, over 15 millimeters. So,
are there phenotypic Schwann cell? I mean, you're taking surals. You got, the whole idea of
that's a very good
sensory
question,
Schwann cell.
and there is a lot written about that.
Yeah,
but I don't know.
But that's not something we're
It's
considering.
not considered. Yeah, because having a population of Schwann cells, you've got all your unmyelinated axons, you've got all your sensory axons in there as well, and they're critical to functional recovery, and they may help maintain nerve structure. So, and I'm personally fairly agnostic at this moment about where the Schwann cells came from, as long as they form like Schwann cells and are able to myelinate. Yeah, it's very, and there's a lot of work on preferential regeneration. We did a lot of work with Tom Burchard, and there definitely seems that there are differences between motor pathways and sensory pathways, which can be recognized by regenerating axons. So I think that is something for the future. So there we are. I mean, there's a lot of work on growth factors. We talked earlier about the first approval of NGF, that its ectopic use for growing unmyelinated axons in the cornea. And there's been a huge amount of work that over the years. Great problem with growth factor delivery is timing and how much is delivered. The Schwann cells are actively handing off these growth factors at exactly the right moment to regenerating axon. Too little, you don't get regeneration. Too much, you also stall regeneration. So it's very, very finely balanced equation. We have some ideas about how to control that in a much more natural way rather than just slow release. And that work's going on in Dublin at the moment. But in conclusion, we originally designed, the original intent for intubulation was as a assist to neuropathy, and I think we've done that. That is fair and available. But however, conduits as they are primarily used for short to medium gaps and not really used because of cost issues, etc., for direct neuropathy. Still, when people are able to sew nerves direct together, they will use direct neuropathy, but they'll also wrap them with various materials. So that's sort of coming on. Then we've talked about matrix field conduits, and they really do accelerate regeneration. But I think more importantly, from my view, is that now it's available in the clinic, that it's the substrate for all the other technology we've been thinking about over the last 20 years. And so you can add ECM components, you can add Schwann cells, you can you can isolate growth factors onto their surface. And I think the hope is good. I mean, 10 or 15 years ago, I don't think we were really very hopeful of much improvement. And now, we demonstrated that with, the dermal regeneration systems regenerate skin better than and more naturally than human autograft. Less scar tissue, it looks more natural. It's absent a few organelles, but it really does do a better job overall. And I think that's something we can look forward to peripheral nerves, at least getting to parity and potentially getting to superiority for long-term catastrophic nerve defects. Thanks.
Thank you very much. The studies of the extracellular matrix components, they were all the channeled graft, correct? None of those would have been the hollow and non-channeled.
Right. So the actual channels are made up of, so it's a bucket of goo poured into the tube, and then the longitudinal freezing is done.
Okay.
All of that work is done by Fergal Brian's team in Dublin through the AMBER Advanced Materials Bioengineering Research Program. I'm governing board member as well.
It is that the Schwann cells take their cue from these extracellular matrix components as to sort of what to do. You should start secreting neurotrophic factors. If that
it does seem to be that that's the case, which is why we're now currently looking for other people are welcome to look as well. Is what is happening after Wallerian degeneration or during the process of Wallerian degeneration and then Schwann cell mitogenesis and then a quiescent phase. There's a lot happening there, which we haven't read from the extracellular matrix, or potentially have, which we haven't really looked at. And so that may actually, creating it will make us go back and look at the natural system and actually get some pointers about maybe there's other potential ECM components which would be of benefit.
The chronicity of it, so over time. If you're doing denervated, so chronically denervated nerve, yes, you've got the chronically exotomized proximal stump and the denervated distal stumps, and the Schwann cells will also be in a very different state.
Yes.
Typically and in numbers and all that sort of thing. And they're not going to repopulate as well.
No, they don't.
Over time, there's probably
it's best to the rate of repopulation just goes down over time. So it's better to immediate repair is always that's what you already from.
So if you've got a chronically axon deprived Schwann cell that you've got 6th or 9th month, and you expose it to these particular.
That's a very good question. What happened? That's an excellent question. Or if you were able to stimulate the Schwann cells in the periphery to maintain their ECM somehow, so that they're ready. And maybe, and that's we know that if you can put guardian neurons experimentally in the distal stump, like small ganglion, which is then inverting the pathway, which keeps the pathway going until you have axons come down from the central.
Have you all been doing that already?
No. Okay. We've been playing.
Still our thunder already. We're working on that right now. It's driven by the IPSC motor neurons, distal neurons do that. So maintain this.
Well, it's so we did a lot of work on the cavernosal nerve in Duke, and we did some collaboration with Patrick Walsh here in Boston, and then we did a couple of grants to look at that more in depth. And the stellate ganglion or pelvic ganglion is really a condensation of, of neurons and autonomic neurons, and you'll find a lot of them in the pathway.
Yeah.
So you'll be going, and you're doing serial sections now. What you think is a peripheral nerve, it's not. It's got neurons, and so it makes it very confounding for models of when you're stimulating, because there's a lot of neurons still in the distal pathway, so
Yes.
you end up stimulating them and have an effect. And so it's the peripheral nerve is not a toxic environment for neurons. You can put neurons into the peripheral nerve. So micro injections of neurons into somatic peripheral nerves seems to be a very viable option.
I think sensory, you put dorsal root ganglion in there. Sensory component, as long as it can create an axon that the Schwann cells can stay around for a while and then they disintegrate, well, and then degeneration, the new nerve comes in. You don't think that they would be, the Schwann cell would be primed to really be accepting sensory rather than.
Well, if you're being really clever, you would produce your own population of neurons, which could dose and kill.
Yes.
And so that they could be a temporary. Roger Madison did a lot of work with chlorine E6 loaded beads in neurons being implanted centrally, and you could illuminate them with a green light, and they would be produce simply oxygen and kill them.
Or you immunosuppress and take it away.
Exactly.
But the sensory versus motor be not something in your mind.
I think it would be ideal, but I think, in my personal opinion, is that maintaining a axonal population in the pathway is the main thing to maintain the Schwann cell population. And also, if you do that and you remove the axons, the Schwann cells will then go through a mitotic division and upregulate themselves again. So it would be by putting the neurons in and then taking them away as you're refreshing the environment.
Yeah.
So
they don't change anyway. Reset.
We had a little bit of this debate with Tessa at a conference last year, which is in an avulsion patient or a spinal cord injury patient. Of course, you lost all of your motor axons, but you get your full complement of sensory axons getting through there. So you should have preservation.
Just demonstrates that you can do that. It's not exclusive. And it's fascinating that you don't have the congruent full sulfate on the outside. And so the axons will penetrate from outside into the nerve much easier than they could come out.
Oh, is that right?
Well, it seems to be. That's a hypothesis, but that's one of the things we can sort of think about.
It's like, I'll tell you, a lot of these are questions that we guess and the answers are no, like when we're putting the tubes on, it should be oversized, it should be undersized, it should be sealed off, should I stick fibrin glue on the ends, and you're telling me that you already know the answer to that question. No fibrin glue required, this thing is going to seal itself.
Yeah, the primates were done single suture each end using exactly that technique long before Integra even existed. So I feel very happy to talk about that. And they were at liberty in their cages immediately so they could hypertrophy fix and brachiate and have one single suture come out.
For the fascicular architecture of the nerve, where you don't have it, or as you did before, so you had all the kind of the compartments, which may be guiding.
That's a great question.
They can't get out of now. You've opened it up.
So that's one of the reasons why when we created the longitudinal matrix, is that if you enter at 2 o'clock position, you exit at 2 o'clock position. Yeah. Each pore is a result of a single ice crystal being grown all the way through the tube, which is why it's relatively complex from a thermodynamics point of view. And Tom Richard, contrary to what was Sunderland's point of view, was that the nerves get completely randomized all the way, up the nerves. Yes. But Tom did some lovely studies of primates where he labeled with HRP from digits, and he found that the group of sensory axons from the digit maintained their position through the entire course, regardless of fascicular pattern.
Within the digital nerves themselves.
No, through the entire course of the median nerve.
Oh, really?
Yeah. So he did sections all the way up the median nerve, I believe median, or I can't remember, and they maintain their position, and there's particles coming and going, and they're all together all the way up. So the vesicular pattern doesn't really matter. It's a rotational alignment, which is critical when you're putting the nerves together. And I think you pointed out that so sciatic nerve in humans is sort of this sort of ribbon of fascicles. It's much wider than it is thick. And so, obviously, if you flip it over and deliberately disconnect everything, you're not going to get very good functional recovery. But if you, connect all the fascicles up, or you connect it in order, you can get appropriate regeneration. So sort of points to how the fascicles are critical for it. But that's a lovely study from Tom. It's just it has images of all the way up the nerve showing that they maintain, they don't wander off.
So you have some particular architecture in there that does guide it into that positioning.
So you have, they'll be coming out. The idea is that you have the nerve of whatever particular pattern has, and you have it, the distal nerve, and the axons will come out, and Schwann cells will come out and enter the tube. The axons are, most of the axons from this side will end up at the other side.
But you just have a Fibrin Clot there without any architecture. And the Schwann cell.
Oh, well,
I mean, with the
talking about the matrix design.
yeah,
with the Fibrin Clots.
you do
tend to
guide
more,
versus
more
no
randomized. But you're still getting a rotation is important.
Yeah.
We always would, we found that, in the primate surgery, even if you sort of lost your space, like if you flooded the field and let the nerve float, it untalks itself and sits perfectly in it like that. And you can look. And the only other thing on that is when we abandoned using saline as a wash for peripheral nerve work, we use lactate, brings lactate. Is that we have far less scar formation. So we worked in the rodent model. So the cavernosal nerve in the rodent is 1.18 millimeters in diameter. So finding it, and then doing intubulation, and then going back for repair, and then going back and doing electrophysiology and retrograde labeling it, and then going back again to find it. If you have any scar tissue, you can't find it. And we found that we used ring and lactase.
Yeah,
it was very clear we could find it each time and actually achieve.
start doing that.
Yeah, I know.
So we have to,
These are nice pointers, and we can hit them every day. We're doing this, obviously standing in the operating room.
and in rats inside the nerve, you saline, you'll get go back in, you'll see a diffuse fibrosis over the entire surface, like a slightly opaque quality. If you use lactate ring, it looks just like normal.
So we're on our way out. One moment. Yeah, thanks.
But,
And now I would have thought that the serum of the body would have washed it and sort of would be irrelevant what you use because
that's what we thought, but it seems to have significant.
There's no studies on this, just an observation.
It's an observation.
we're going to break like first patient.
But I'm always open to doing somebody picking it up and doing a study or something when I'm retired.
So since they're equally available, and now that I've heard this, there's no reason that I wouldn't switch with a lactate rigor in the operating room.
And the thing is, what took us there was that we were doing rapid fusion with phosphate buffer saline, and it stopped the heart.
Yeah.
And so we started using ring lactate, and the heart beats throughout. It's a physiological solution. Okay. And the heart beats throughout the perfusion, you get a fantastic perfusion. And so we had lots of cases of ring lactate in the lab and no saline. We saw no reason why we shouldn't use ring as lactate. And then noticed that it was so much easier to do all the operations, particularly for capitalism. Interesting. So definite serendipity.
Yeah.
Thank you very much.
No, thank you very much. We've been